What if your skeleton was a passport, a chemical diary, recording every place you've ever lived, every major change in your diet, every long-distance journey you ever took? It's not science fiction. Deep within our bones and teeth lies a hidden chronicle, and today, we're going to learn how to read it. This is the world of forensic isotope analysis, a field that is transforming silent skeletons into storytellers and allowing us to reconstruct the epic journeys of our ancient ancestors. The fundamental idea is surprisingly simple. You are what you eat and drink. Throughout your life, the elements from your food and water, from the local geology and rainfall, are absorbed and locked into your tissues. The specific chemical signatures of these elements become a permanent record. By analyzing this record, scientists can tell the life story of an individual who lived thousands of years ago. At the heart of this are isotopes. Remember from chemistry class, isotopes are just different versions of the same element. They have the same number of protons, but a different number of neutrons, which gives them a slightly different weight. Think of them as different regional accents for the element. For example, the oxygen in the water you drink has a different isotopic accent in a cold, high-altitude mountain range than it does in a warm coastal region. Scientists use an incredibly sensitive machine called a mass spectrometer to measure the precise ratio of these different isotopes in a sample of bone or tooth. They then compare that chemical fingerprint to global maps of these isotopic signatures, called isoscapes, to pinpoint where that person might have lived. But the real power comes from using a whole toolkit of different elements, because each one tells a different part of the story. Let's start with the geographic detective, strontium. The ratio of strontium isotopes in the ground is determined entirely by the age and type of the underlying rocks. As those rocks weather away, their unique strontium signature gets into the soil, the water, and eventually, the plants and animals. When a person eats local food, that strontium is built into their skeleton. Here's the brilliant part. Tooth enamel forms in childhood and never changes. It's a snapshot of where you grew up. Bone, on the other hand, is constantly remodeling itself. The strontium in your bones reflects where you've been living for the last several years of your life. So, if a skeleton's teeth have a strontium signature from the mountains, but its bones have a signature from the coast, you've just found definitive proof that this person migrated. Next up is oxygen. The oxygen isotopes in your body come mostly from the water you drink. As we said, the isotopic signature of rainwater changes with climate, altitude, and distance from the sea. So the oxygen in your teeth and bones provides another, independent line of evidence to track where you've been. It's like having a second GPS tracker confirming the data from the first. But where you're from is only half the story. What did you eat? For that, we turn to carbon and nitrogen. These are the storytellers of ancient diets. The ratio of carbon isotopes can tell us what kinds of plants someone ate. Plants are broadly split into two groups, C3 and C4, based on how they do photosynthesis. C3 plants include things like wheat, barley, and rice. C4 plants include maize, millet, and sugarcane. They have completely different carbon signatures. So, if we find a skeleton in a wheat-growing region, but its carbon isotopes show a lifetime of eating maize, we know that person came from a maize-based culture. It's another powerful clue for migration. Nitrogen isotopes tell us about your position in the food chain. The higher up the food chain you go, the more of the heavier nitrogen isotope you accumulate. So a herbivore has a low nitrogen signature. A carnivore that eats herbivores has a higher one. A carnivore that eats other carnivores has an even higher one. A diet rich in marine fish also has a very distinct, very high nitrogen signature. By combining these elemental clues, archaeologists can build what they call an isobiography, a detailed life history of an ancient individual. It's one thing to talk about the theory, but let's see how it solves some of the past's greatest cold cases. First up, Ötzi the Iceman. In 1991, a stunningly preserved 300-year-old body was found melting out of a glacier in the Alps. For years, his origin was a mystery. Was he from the Austrian side of the mountains or the Italian side? Isotopes gave us the answer. Analysis of his tooth enamel, his childhood signature, showed oxygen and strontium values that pointed to a specific valley south of where he was found, the Isotope Valley in modern-day Italy. But the isotopes in his bones, his adult life, and in tiny flecks of mica from his food found in his gut, told a different story. They matched a different valley, the Vinci, also in Italy. This wasn't just a man who died in the mountains. This was a man who had moved during his lifetime, from one community to another. The analysis even revealed his last meal, ibex and red deer meat, with einkorn wheat. We know his life story in a way that was unimaginable just a few decades ago. Now for case file number 2, the Amesbury Archer. Discovered in 2002 near Stonehenge, this was one of the richest Bronze Age burials ever found in Britain. He was buried with over 100 artifacts, including gold hair ornaments and some of the earliest copper tools in the region. This was clearly a very important, high-status man. But who was he? Oxygen isotope analysis of his tooth enamel delivered a bombshell. He was not a local. His chemical signature showed he grew up in a colder climate, most likely the foothills of the Alps in Central Europe. This was groundbreaking. It was the first direct evidence that the new technology of metalworking wasn't just traded into Britain, it was carried by people, by immigrants who traveled vast distances. He was a pioneer, bringing revolutionary ideas to a new land. And adding another layer to the story, a second man buried near him, known as the Companion, had an isotopic signature consistent with a local British upbringing. This paints a fascinating picture of newcomers and locals interacting and integrating thousands of years ago. For our final case, let's zoom out from an individual to an entire city. Teotihuacan, in central Mexico, was one of the largest and most influential cities of the ancient world, bigger than Imperial Rome at its peak. Was it a homogeneous empire, or something else entirely? Researchers analyzed remains from different neighborhoods all across the city. Using strontium and oxygen isotopes, they discovered a vibrant, multi-ethnic metropolis. A huge portion of the population were migrants, hailing from as far away as the Maya region, hundreds of miles to the east. In one district, it's estimated nearly half the population were immigrants, and the diet isotopes, carbon and nitrogen, showed a complex social fabric. Some migrant groups clung to the diets of their homelands, a form of cultural retention. Others assimilated, adopting the local Teotihuacan diet, which was heavy on maize. High-status individuals, and even sacrificial victims buried in the Great Pyramids, were also found to be foreigners. This tells us that migrants played complex and important roles at every level of society in this ancient cosmopolitan center. Now, as powerful as this technique is, it's not magic. One of the biggest challenges is the need for good baseline maps, or isoscapes. If you don't know the isotopic signature of a particular region, you can't match a person to it. Also, the diet can sometimes complicate things. A person living on the coast who eats a lot of marine fish might have their local strontium signature masked by the strontium from the ocean, and there are significant ethical concerns. The analysis is destructive. It requires removing a small sample of bone or tooth. These aren't just artifacts. They are human remains, and the decision to perform such analysis must be done with great care and respect, often in consultation with descendant communities. So what does the future hold for this incredible field? The technology is constantly improving. New laser techniques allow scientists to analyze microscopic parts of a tooth, giving a week-by-week timeline of someone's childhood movements. But the most exciting frontier is the integration of isotope analysis with ancient DNA analysis, or aDNA. Isotopes tell you where a person lived and what they ate. their geographic and cultural story. ADNA tells you about their genetic ancestry. Combining them allows us to disentangle complex questions of identity. For example, did a person with local ancestry move away and then return? Or was this a person with foreign ancestry who moved into the region for the first time? Together, these two techniques provide a biography with a richness we've never had before. The whispers from the bones are growing louder and clearer every year. We are learning that skeletons are not silent objects, but dynamic records of individual lives, filled with epic journeys, cultural choices, and personal histories. As the science continues to advance, we can expect to uncover even more intimate and compelling stories, adding countless new chapters to the saga of what it means to be human. Thanks for tuning in to G Fun Facts Online. Join us next time as we uncover more incredible stories from the world of science.